Hello. Good evening to the student who have joined. Can you let me know in the chat box if my voice is audible to you? And also let me know if you can see the video and presentation. Okay, thank you for the confirmation. We will wait till it's 6 p.m. and then we will start today's session. Okay. Meanwhile, please kindly wait. Okay, so it's almost 6 p.m. I think I should start with the introductions, and I have made the call open. So if other students want to join at a time, they can join. Okay, let me wait for a minute, then I will start. Okay, let me start with the session. So, very good evening to the students who have joined. My name is Mousumi Deb and I'm the PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL course of Organic Chemistry and Biology and Drug Development. And today it is our week 11 live session. So, as you know, every week we do this kind of problem solving session from Monday from 6 p.m. to 8 p.m. And every week we solve the course content of that particular week, which is live at that time. So right now, today it is the last week's means the week 12 course content. This course content is your final course content of this organic chemistry and biology and drug development. We will not have any more new topics after today's class. Okay. So after today's class, we will have today's whole 11 live session. We will have two more live session. One will be on next Monday, which will be a kind of summary class where it will be half of type of class. Mine means in half class I will solve few questions, and in other portion, other half time, I will discuss few topics which are important for your this course, organic chemistry and biology drug development, which I couldn't give emphasis on earlier, and some topics which students have told me to cover up. So those topics I will discuss on in the next class, okay, which will be in the I think on the 14th October in our 12th week live session. So that class will be a half of class, one question answer, and also the summary, which I will teach you some few topics. But today in this week 11 live session, I'm gonna discuss about the course content which are present in your week 12. Okay, so this is the last course class where we will have some new topics. So before start, I will like to know that if my voice is audible to you, please let me know if there is any issue with my voice and also the with the video. Kindly let me know. So before starting, I would like to point out few things that there will be this kind of problem solving session every week. Today it is our week 11 live session. We will have one more live session in next Monday from 6 p.m. to 8 p.m. And the last live session will be on Wednesday, which is 16th of October, and that will be a summary session. Okay, there won't be any question answer, but I will summarize few things from your course which are necessary and which will be beneficial for your final examination also. So these sessions will be live on the Google Meet platform, as I have already mentioned. So if any network disruption happens from my side, please do not leave the class immediately. Okay, please wait. I will try to join as soon as possible. And during the session, the students can post their question and doubts through the chat box. Since I am recording the session, I have muted your microphones and also the cameras so that I can record it smoothly. Okay, so if you have any doubt, any query, you can let me know through the chat box. The chat box is always open, and I'm monitoring the chat box every now and then. So you can let me know if any problem, any doubt, anything. You can let me know through the chat box. But if still any doubt persists after class, you can drop an email to me. I will answer your queries through email also. I will drop my email ID at the end of the session. This presentation which I am showing to you and the class recording of today's class. Okay, these two things we will sharing with the students, and I hope you know where to find this kind of recordings. Okay, so just let me show you where to find this problem solving sessions recording. So first you have to go to the organic chemistry in biology and drug development in the SWAM portal. Okay, then when you log in to the SWAM portal, what you will see, just let me show you one time. Once you log in, you will find there is an announcement tab at the left portion. This is your course outline. Okay, here you will find there is a problem solving session, July 2024, and you are here. You will find the problem solving session recording. Okay, if you click on that portion, you will be redirected here, and here you will find all the YouTube links for the previous sessions, and this Google Drive folder. You will find all the things which are all the all the PPTs which I have told you. Okay, the final examination, the pattern, it will be subjective, objective both. Okay, there not only there will be a small, I mean MCQ type of question, there will be subjective answers which you have to write. So the it will be a mixed pattern. It's not only the MCQ, but it will have subjective, objective both. So you have to be prepared for the both things. Okay. Since the final examination have the subjective things, whenever I am doing the MCQs only in the class, but when I am showing you answer, I am discussing the topic in detail so that you can write a detailed paragraph on the particular topic also. Okay. So let us start with today's session. In today's session, I have already said that it is the week 11 live session where I am going to discuss the week 12 course content. So let us see first what are in the our week 12 course content of the organic chemistry, biology and drug development. So these are the topics for the week 12. We have the cancer and chemotherapy followed by anti-cancer drugs. We have aromatase inhibition and anti-ulcer drugs. We have cholesterol lowering agents. We have cholesterol biosynthesis. We have pharmacokinetics, pharmacodynamics, and QSAR principles. So these are the topics which are very important topics for medicinal chemistry and organic chemistry in biology and drug development. This topic are very, very important. So now let us start. Let us with the first question of today's session. So the first question of today's session is, it says that which of the following is not a microtubule inhibitor? Your options are number A cisplatin, number B paclitaxel, number C vinblastine, and number D colchicine. So which of the following is not a microtubule inhibitor? Cisplatin, paclitaxel, vinblastine, and then colchicine. I will wait for 2 to 3 minutes. Then I will check the chat box and then I will show you the answer and we will discuss it in detail. I will discuss it in that manner so that if you are set to write a note on microtubule inhibitor, you can do it. So I will show you the answer like that way. So first identify which of the following is the correct answer. Which of the following is not a microtubule inhibitor? Cisplatin, paclitaxel, vinblastine, colchicine. Which of the following is not a microtubule inhibitor? Cisplatin, paclitaxel, vinblastine, colchicine. I will be checking the chat box soon, so please let me know A, B, C, or D. Even if your answer is wrong, it's fine, but let me know which of the following you think is the correct answer. Okay, let me check the chat box now. Okay, I find there is few answers. It is not there is a particular pattern. It is depending on how we are setting the examination question. Okay, there is no particular, I mean, performer. It can be anything. That's what I can say. Okay, so I have a few answers. I have two now, D and one C. Okay, let me check which of the following is the correct one, C or D. So the correct answer, the agent which is not a microtubule inhibitor is, it is number A, cisplatin. So cisplatin is not a microtubule inhibitor, but others like paclitaxel, vinblastine, colchicine, these are the microtubule inhibitors. Okay, so this is a chart where there is a different microtubule inhibitors. We have paclitaxel, which is a, or with this is the NAP paclitaxel. Their binding domains are taxane binding. The treatment for breast, ovarian, prostate, lung cancer, and mode of action is the anti-microtubule depolymerization leading to mitotic arrest. Okay, before going to this chart, it will be better if I go to this portion. So there are microtubule inhibitors, are the drugs which target the tubulin. Okay, so first let us understand what are the tubulin. So tubulin are present and very important during the cell division. Okay, the cell division, this tubulin are very important. So there are different drugs which target the tubulin as a part of the anti-cancer activity. So first are the vinca alkaloids. So vinca alkaloids are a type of natural products which binds with the tubulin, inhibit the microtubule formation. So these are the these are the vinca alkaloids. This yellow dots, these are the vinca alkaloids. No, this is the this is the tubulin, and this cyan color V-shaped portions are the vinca alkaloid. So they bind with this tubulin and inhibit the microtubule formation. That is how they perform the metaphase arrest, and they show the anti-cancer property. Metaphase is a phase in the cell division. We all know there are four phases like prophase, metaphase, anaphase, and telophase. So metaphase arrest is done by this particular vinca alkaloids and by binding with the tubulin, so that it can inhibit the microtubule formation, and that is how it exerts its anti-cancer activity. And there are examples are the vincristine and vinblastine. These are the vinca alkaloids which are a microtubule inhibitor. Okay, so these are all the, now we have another example of which are the taxanes. So taxanes are the compounds which actually they bind with, freezes the microtubules in the metaphase, and tubulin molecules fail to repolarize. Okay, so first the microtubule formation is inhibited by this vinca alkaloid, but after formation of the microtubules, this taxane, they freeze microtubules in the metaphase only. Okay, and if the depolymerization do not happen, so how which happen, not anaphase, so anaphase won't happen if depolymerization don't happen, so it will lead to the cell death, and that's how it exerts its anti-cancer activity. So metaphase arrest is done by this vinca alkaloid, and freezing of the microtubule after formation means inhibiting the tubulin molecules from depolarization is done by taxanes. Then we have colchicine. Colchicine binds to tubulin, inhibit the microtubule polymerization, impaired activation and migration. And we have neutrophils, this reduce inflammation in the gout. Then we have the griseoflavin. This binds to the fungal tubulin, impaired microtubule formation, inhibit mitosis and fungostatics. And then we have the mebendazole and albendazole. These are the blocks polymerization of tubulin in intestinal cells, blocks the glucose and glucose uptake, and gradual immobilization and death in Helminth. So these are the different agents which are not only as a part of an anti-cancer drug, but different top types of diseases, and these are the examination which are particular in case of the anti-cancer ones. So there are different examples. Then I will just again I will tell you. So microtubule inhibitors, as I mentioned, it's paclitaxel. It is a taxane binding which binds here. And cancer treatment for breast, ovarian, prostate, and lungs. Mode of action: anti-microtubule depolymerization lead to mitotic arrest. Then we have the docetaxel. So this is a taxane binding, breast, non-small cell lung cancer, androgen independent metastasis, prostate cancer. The mode of action: anti-microtubule depolymerization and attenuation of BCL2 and BCL-XL gene expression. We have colchicine, which is colchicine binding, and the cancer treatment for hepatocellular and prostate cancers. This is anti-microtubule polymerization, its mode of action and cell cycle arresting metaphase. And vinblastine are the vinca alkaloid. In the cancer treatment are testicular, Hodgkin's and non-Hodgkin's lymphoma, breast and germ cell cancers. And mode of action induces the wedge between the tubulin interface, causing tubulin self-association into spiral aggregates, antimicrotubule polymerization, and cell cycle arresting metaphase. So these are the microtubule inhibitors along with their binding domain, cancer treatment, and mode of action. Okay. These are the drugs which target tubulin, and they lead to the different feature different clinical aspects. So we have cisplatin. So cisplatin is main mechanism of the cytotoxic action involves the binding of cisplatin to the genomic DNA and in the cell nucleus to form intrastine and intestine post lining. Okay, this interferes with the normal transcription or the DNA replication mechanism and triggers the cytotoxic process that lead to the cell death. So this is the mechanism of action of cisplatin, which is not a microtubule inhibitor. So the correct answer was number A, cisplatin. Okay. So now I will go to the next problem. Problem number 2 says chlorgyline is the specific inhibitor of dash. You have to fill in the blanks. So your options are number A, myo B, number B, myo A, number C, acetylcholinesterase, and number D, ACE. Chlorgyline is a specific inhibitor of which enzyme? Myo A, myo B, acetylcholinesterase, or ACHE, or ACE, which is angiotensin converting enzyme. You have to find out chlorgyline is a specific inhibitor of which particular enzyme. Again, I will give you some time. Think carefully and then let me know in the chat box A, B, C, or D which of the following is the correct answer. Chlorgyline is a specific inhibitor of which enzyme? Let me check the chat box. Okay, I have one answer which says it's number B. Okay, let me check if there are any other answers. Okay, let me check which of the following is the correct answer. So the correct answer is yes, it's number B, myo A. It is a specific chlorgyline is a specific inhibitor of myo A. This is the structure of the chlorgyline, which has this. Okay, let me open the laser pointer, which has this 1,3-dichloro with this chain where there is a triple bond at the terminal position also. So myos means monoamine oxidase inhibitors. Okay, so monoamine oxidase inhibitors or myoice had a huge class of powerful antidepressant drugs. They are particularly effective in treating atypical depression, panic disorder, and social phobia. Due to potentially lethal dietary and drug interaction, MyoIce have been reserved as a last line of RX, means which are only when it is used only when other classes of antidepressant drugs like SSRIs and TCS have failed. There are two types of isotypes of enzymes. One is the myo-A enzyme, which is present in the cytoplasm of the neurons of CNS and the peripheral tissues, for example, liver. It acts as a non-specifically on Na5-HT and dopamine. Chlorgyrin is a specific inhibitor of myo-A. Then we have myo-B enzyme, which is present mainly in the CNS and acts more on the dopamine. Serotonin is a specific inhibitor of myosin, and paraglin is a non-selective MOAI. Okay. So this is just remember chlorgyrin. It is a specific inhibitor of myoA, and these are the myoids, which are the typical which treats the depression, but it is have a very lethal action on the body, so that it is only used as a last resort of antidepressant drug. Okay, there are other classes of antidepressant like SSRIs and TCS also. So now I will go to the next problem. Problem number 3 says identify the correct statement about the mitomycin C. Your options are number A, it contains an azitidine ring. Number B, it is a prodrug. Number C, the first step of in its method of action is an oxidation by NADPH. And number D, it forms a pyrazolo hydroquinone as an intermediate. Identify the correct statement about mitomycin C. Number A, it contains an azitidine ring. Number B, it is a prodrug. Number C, the first step in its method of action is an oxidation by NADPH. And number D, it forms a pyrazolo hydroquinone intermediate. Which of the following is the correct statement about mitomycin C? Number A, it contains an azitidine ring. Number B, it is a prodrug. Number C, the first step in its method of action is an oxidation by NADPH. Number D, it forms a pyrazolo hydroquinone intermediate. Let me check the chat box now. Okay, I have one answer which says it's number A. Okay, let me check if number A is the correct answer or not. So the correct answer is, it is number B. Okay, the correct statement about mitomycin C is number B, which says that it is a prodrug. Now let us first discuss about, this is the structure of the mitomycin C. Okay, so the mitomycin is converted after, in the first step, okay, of the method of action is the formation of the mitomycin hydroquinone. So as you can see, the first step is there is a enzyme which is which acts in the first step. It is the NADPH or CYP45 reductase. Okay, so the first step of its method of action is the oxidation. It is the reduction by NADPH or CYP45 reductase. Okay, so as you can see here, there is a hydroquinone, there is a quinone structure which is converted to hydroquinone, means the reduction happens and in the first step. Okay, so that is why the statement number C is incorrect. Now for the second step is the formation of the indole hydroquinone intermediate. Okay, this structure is known as the indole hydroquinone. This is the hydroquinone structure and there is a indole ring also. So it is known as the indole hydroquinone intermediate. So it the number D statement which says it forms a pyrazole hydroquinone intermediate, it is also incorrect. Then followed by the binding with DNA. Okay, so this mitomycin DNA adapt form adapt which cross-link DNA, and this here also we will have a Fenton type of reaction which generates hydroxyl radicals which are cytotoxic, and that is why how this mitomycin C show its method of action. So now we have two statements, and now first step is the statement number B, which says it is a prodrug. It is the correct one because this is not a active structure. This is a prodrug which is converted to the active drug by through via two step. Okay. And then it's number one statement which says that it has the azatidine ring. This is incorrect because azatidine ring is a four-member ring which has a nitrogen. But here we have a three-member ring which have a nitrogen, and this name is the aziridine, not azatidine. Okay, so that is why problem the statement number A is also false. So the correct statement is number B, which says it is a prodrug. So remember, in the heterocyclic rings, especially nitrogen containing heterocyclics are very important. So please try to remember this. Now I will go to the next problem. Problem number 4 says identify the statement that is incorrect. Your options are number A, DNA synthesis checkpoint exists in the growth phase of the cell cycle. Number B, in the synthesis phase, the DNA synthesis checkpoint is present. Number C, cancer can result when one or more cell cycle checkpoints malfunction. And number D, G2 phase occurs in between S and M phase. You have to identify the statement which is incorrect. Number 8, DNA synthesis checkpoint. Synthesis checkpoint exists in the growth phase of the cell cycle. Number B, in the synthesis phase, the DNA synthesis checkpoint is present. Number C, cancer can result when both one or more cell cycle. Okay, 1 second. Okay. Hello. So is my voice audible? Let me know in the chat box if my voice is audible to you or not. Okay, sorry, there was a internet issue, so I hope my voice is audible. Wait, let me just share the screen once more time. Okay, So now this is visible to you, right? The presentation. Let me know in the chat box. Let me know if the presentation is visible to you. Okay, okay, fine. Sorry, so there was a issue with the internet. So I will repeat the question number A4. Okay. So problem number 4 says identify the statements that is incorrect. Your options are number A, DNA synthesis checkpoint exists in the growth phase of the cell cycle. Number B, in the synthesis phase, a DNA synthesis checkpoint is present. Number C, cancer can result when one or more cell cycles checkpoints is malfunction. And number D, G2 phase occurs in between S and M phase. So which of the following statement is incorrect? Number A, DNA synthesis checkpoint exists in the growth phase of cell cycle. Number B, the synthesis phase. In the synthesis phase, the DNA synthesis checkpoint is present. Number C, cancer can result when one or more cell cycles checkpoint malfunction. Number D, G2 phase occurs in between S and M phase. A, B, C or D, let me know which of the following is the correct. A, B, C or D, which of the following is the correct answer? Let me know. Okay, let me check the chat box now. Let me see if there are any answers for problem. Okay, I have one answer which says it is the number B. Okay, let me check if number B is the correct answer or not. So the statement which is incorrect is, it is number A. Okay, DNA synthesis checkpoint exists in the growth phase of the cell cycle. This statement is incorrect. Now, let me show you what is the cell cycle. Okay, so this cycle is the cell cycle, and the different checkpoints which is the present in the cell cycle. So this diagram describes the different checkpoints in cell cycle and the responsibilities of each cell cycle. So first is the cell growth cycle. Okay, the cell growth cycle or G1 cycle, and there is a checkpoint. Okay, so this occurs toward. So first is the cell growth checkpoint, which is known as the G1. So this occurs toward the end of the growth phase 1. Okay, G1 is the growth phase 1, and the cell growth checkpoint occurs at the end of this G1 phase. So it checks whether the growth cell is big enough and has made proper proteins for the synthesis phase, and if not, the cell goes through a resting period, which is G0, until it is ready to divide. Now followed by there is the DNA synthesis checkpoint, which occurs during the synthesis phase or S phase. It checks whether the DNA has been replicated correctly. If so, the cell continues to mitosis. Then we have a growth phase 2, where it checks if the cell is properly ready for the mitosis, and we have the M phase or mitosis phase. So here it checks whether the mitosis is complete, and if so, the cell divides and the cell cycle repeats. So there are different checkpoints. Okay, so first checkpoint is present at the end of the growth phase 1. Now, which is a cell growth checkpoint, followed by a DNA synthesis checkpoint, which is present in the S phase, and there we have a mitosis checkpoint, which is present in the M phase. So the statement number A, which says that DNA synthesis checkpoint exists in the growth phase, which is incorrect, because DNA synthesis checkpoint exists in the S phase or synthesis phase. Basically, stalled DNA replication fork will activate the S-phase checkpoints where it is ensured a proper replication of the genetic material. If this checkpoint is somehow missed, the another checkpoint at G2 phase re-ensure proper replication devoid of any DNA damage prior to initiating the mitosis cycle. And so there are different stop points. Okay, first G1 phase is the stop point is the damaged DNA. If DNA is damaged, it is stopped unreplicated, and damaged DNA stops the phase. Then unreplicated damaged DNA stops at the end of the G2 phase also, and chromosome misalignment stops the cell cycle in the M phase at the end of the M phase. So there are different stop points. If there are this kind of defects, the cell cycle will not proceed further. Okay. So cancer actually happens due to different malfunction of the checkpoints of a particular cell cycle. So problem number 4, the correct answer was number A. Now I will go to the next problem. So problem number 5 says, what is the mode of action of proflavin? Your options are number A, alkylating agents, number B, chain cutting agents, number C, antisense agents, and number D, intercalating agents. So what is the mode of the action of proflavin? Again, I repeat, what is the mode of action of proflavin? Number A, and chain cutting agents. Number A, alkylating agents. Number B, chain cutting agents. Number C, antisense agents, and number D, intercalating agents. What is the mode of action of proflavin? I will wait for 2 minutes, then I will check the chat box and show you the answers. Let let me know which of the following you think is the correct answer. What is the mode of action of proflavin? Number A, alkylating agents. Number B, chain cutting agent. Number C, antisense agent, and number D, intercalating agents. Let me check the chat box now. Okay, so let me show you the answer for this problem number 5. So the correct answer is number D, intercalating agent. So proflavin acts as an intercalating agent, and its mode of action of is the intercalation. So intercalating agents, example proflavin. So these are the drugs which acts on the DNA itself. So this is the structure of proflavin. As you can see, there are three rings which are fused with each other, and this has a very planar structure. And at the end of the structures, it has the ammonium ions, which are the positive charge. So this particularly the proflavin, it acts this planar structure of proflavin. It acts with the sugar. It acts with the base pairs of the DNA, and the terminal ends. The positive charge. It interacts with the negative charge in the sugar backbone of this particular DNA. Okay, so this wavy bonds, it notates the van der Waals interaction. Okay, the planar structure is interacting with the van der Waals interaction with the DNA base pair, and ionic interaction is shown by this dot line. So which is the ammonium ion, and the O minus acts with the ionic interaction. So chemical mutagens, intercalating agents contain flat planar structures that intercalate themselves into the double helix. This distorts the helical structure. At replication, a template that contains an intercalated agent will cause insertion of a random extra base. The base pair addition is complete after another round of replication, during which the intercalating agent is lost. For example, ethidium bromide, proflavin, acridine orange. So these are the intercalating agents. So intercalating drugs are the compounds that contain a planar or heteroaromatic features which slip between the base pair layers of the DNA double helix. Some of these drugs prefer to approach the helix via a major groove and others prefer access via the minor groove. Once they are inserted between the nucleic acid base pairs, the aromatic and heteroaromatic rings are held there by the van der Waals interaction with the base pairs above and below. Several intercalating drugs also contain ionized groups which can interact with the charged phosphate groups in the DNA backbone, thus strengthening the interaction. Proflavin is an example of a group of antibacterial compounds called aminoacridines, which were used during the World Wars I and II to treat surface wounds. These drugs proved highly effective in preventing infection and reduced the number of the fatalities resulting from the wound infection. Proflavin is completely ionized at a pH 7 and interacts directly with the bacterial DNA. The flat tricyclic ring intercalates between the DNA base pairs and interacts with them with the van der Waals forces, while the ammonium cations in the ionic bonds are negatively charged phosphate groups on the sugar phosphate backbone. Once inserted, proflavin deforms the DNA double helix and prevents the normal functions of replication and transcription. Okay, so this is the mode of action of proflavin. Now I will go to the next problem. I hope it is clear. So till now we have 5 questions till now in today's session. Do you have any problem in any question? Please let me know. If you have any doubt, any query, you can let me know here. Otherwise, I will move to the next problem. Okay, so let me move to the next problem of today's session. So problem number 6, it says that which cyclin is involved in balancing cell growth and division? Your options are number A, cyclin A, number B, cyclin B, number C, cyclin D, and number D, cyclin E. Which cyclin is involved in balancing cell growth and division? Cyclin A, B, C, cyclin A, cyclin B, cyclin D, or cyclin E? Which cyclin is involved in balancing the cell growth and division? Please let me know. Even if your answer is wrong, it's fine. But let me know in the chat box which of the following is the correct answer. Which cyclin is involved in balancing cell growth and division? Cyclin A, B, D, or cyclin E? Which cyclin is involved in balancing cell growth and division? Cyclin A, B, D, or E? Okay, let me check the chat box. Hello, is my voice audible now? I think there is a internet issue. Please let me know if my voice is audible now. Okay, so there is an internet issue, so I think that is why the voice is breaking sometimes. So let me know if there is any issue again. Okay, I'm trying to change the mode of the internet also. So please let me know. Anyways, I am recording the session, so I hope it will be fine with the recording. So please bear with me. Okay, so I have one answer for this problem which says it is number D. Let me check if number D is the correct answer or not. So the answer for problem number 6, which says that which cyclin is involved in balancing the cell growth and division, it is number C, cyclin D. So cyclin D is involved in balancing the cell growth and division. So there are the different cyclins which are present in the cell cycle, and the cyclin and the cyclin. So control of the cell cycle by cyclins and cyclin dependent kinases are shown here. So there is CDK, the cyclin dependent kinase 4 and 6 in the G1 phase, and cyclins D1, 2 and 3. Then we have the CD2, CDK2, which is cyclin dependent kinase 2, and cyclin E in this at the end of the G1 phase. We have CDK2, or the cyclin cyclin dependent kinase 2 and cyclin A in the S phase, and cyclin dependent cyclin A in the G2 phase, and cyclin B in the M phase. The control of the cell cycle involves in variety of proteins called cyclins and the enzymes known as the cyclin-dependent kinases or CDKs. There are at least 15 types of cyclin and 9 types of CDK, and each has a role to play at different stages of the cell cycle. Examples are shown here in the figure. The binding of the cyclin with its associated kinase activities activates the enzyme and serves to move the cell from one phase to cell cycle to another. For example, when a cell is in the G1 phase, the decision has to be made whether it is to move into S phase and start copying DNA. This decision is taken by depending on the balance of the stimulatory versus inhibitory signals being received through the signal transduction. If the balance is toward the cell growth and division, there is an increase in the cyclin D. This binds with the CDK4 and 6, and resulting complexes phosphorylate a powerful growth inhibitory molecule known as PRP, which normally binds and inactivates the transcription factor. From phosphorylation alters the PRB such that it can no longer bind with the transcription factor, and the latter is free to bind with the specific regions of DNA. This results in the transcription and the specific genes, further leads to the production of the proteins capable of moving the cell toward the S phase. Cyclin E and the thymidine kinase. Once cyclin E is produced, the combines with the CDK2, and the complex is responsible for the progression from G1 to S phase. Other activated cyclin CDK complexes are important in the different phases of cell cycle. For example, cyclin A CDK2 complex is required to progression through the S phase, and cyclin B CDK1 complex necessary for the mitosis. So the cyclin which is mainly involved in the balancing the cell growth and division is the cyclin D. So number C was the correct answer. Is my I hope it is fine with you now. My voice is clear. If there is any issue, please keep letting me know. Okay, if there is any issue with the voice and also with the presentation, keep letting me know. So this was the problem number 6. Now I will go to the next problem. So problem number 7 says. So problem number 7 says, which DNA base interacts with the anti-cancer intercalating agent dactynomycin? Your options are number A adenine, number B thymine, number C cytosine, and number D guanine. Which DNA base interacts with the anti-cancer intercalating agent dactynomycin? Adenine, thymine, cytosine, or guanine? Again, I will wait for some time. Which DNA base interacts with the anti-cancer intercalating agent dactynomycin, adenine, thymine, cytosine, guanine? Which DNA base interacts with the anti-cancer intercalating agent actinomycin? Let me check the chat box. Okay, I have one answer which says it's number C. Okay, let me check if number C is the correct answer or not. So the correct answer for problem number 7 is it's number D, guanine. So guanine is a DNA base which interacts with the anti-cancer intercalating agents dactinomycin. So this is the structure of the dactinomycin. So another example of intercalating agent is dactinomycin. It is previously called as actinomycin D. It is naturally occurring antibiotic that is first isolated from the Streptomyces, and it has been shown an effect as the effective as have an effect as an effective anti-cancer in childhood cancer. If you look at the structure of dactinomycin, there is a planar phenoxazone system. This is the phenoxazone system, and it is also has a peptide over here, which is a cyclic peptide. Here it is has a cyclic peptide also. This drug approaches DNA via the major group of the double helix, and after it intercalates the planar tricyclic system, this one grows in between two base pairs, as we have seen previously. The charged amino acid group attached with the sugar is important. Is also important and forms an ionic bond which is negatively charged in the phosphate group of DNA backbone. So as I have mentioned, this portion is positively charged, which interacts with the negatively charged sugar backbone of the DNA, and this planar structure slides between the DNA base pairs and interacts through the van der Waals interaction. It appears to favor interaction with the guanine cytosine base pairs as GC base pairs, in particular between two adjacent guanine bases on the different on the alternative strand of the helix. So you have GC interaction and you have two Gs, then it prefers to bind with this type of interaction base pair. Molecule is further held in position by hydrogen bond interaction between the nucleic acid bases and cyclic pentapeptides, which we have looked earlier. The two amino groups of the guanine plays a particularly important role in the interaction. The resulting bound complex is very stable and prevents the unwinding of the double helix. So, in this turn, prevents the DNA-dependent RNA polymerase from catalyzing the synthesis of mRNA, and thus it prevents the transcription. So, the effect of dactinomycin is actively dividing cell is once goes inside and does the intercalase. After it intercalates, it forms extremely stable bond with DNA, and after this complex is formed, because of this complex is so stable, it prevents the unwinding of the double helix. And if unwinding of double helix does not happen, as we know from previous lectures, that the single stranded DNA is not produced, which is going to transfer the information to mRNA. So therefore transcription is inhibited, and as the consequences of this inhibited transcription, the cell will stop replicating and stop dividing, and that is how the anti-cancer property of this dactinomycin is produced. Okay. Now I will go to the next problem. So problem number 8 says method of action of aromatase inhibitor is, your options are number A, irreversible inhibition, number B, reversible inhibition, number C, suicide inhibition, and number D, uncompetitive inhibition. So which of the following is the correct mode of action of aromatase inhibitor, or as I should say that the method of inhibition of aromatase inhibitor is irreversible inhibition, reversible inhibition, suicide inhibition, or uncompetitive inhibition. Let me know A, B, C or D, which of the following is the correct answer for problem number 8. The method of action of aromatase inhibitor is number A, irreversible inhibition, Number B, reversible inhibition. Number C, suicide inhibition. And number D, uncompetitive inhibition. Method of action of aromatase inhibitor. So this different inhibition we have also learned earlier, that about irreversible, reversible, suicide, and uncompetitive inhibition, and about the different inhibitions I will discuss again in the our summary class, which is due on the next week. So what is the method of action of aromatase inhibitor? Okay, let me check the chat box now. Okay, I have one answer which says it is number C. Let me check if number C is the correct answer for this problem number 8. So the method of action of the aromatase inhibitor is very good. It's this number C, suicide inhibition. So what is first? Let us remind once more what is remember once more what is the suicide inhibition. So in suicide inhibition, a suicide inhibitor initially is inactive, attached to an active site of an enzyme and undergoes catalysis to form a product which occupies the active site and thus covalently binds to it. This binding alters the enzyme in such a way that enzyme is damaged irreversibly. So first, we have a suicide inhibitor binds with the particular enzyme, and suicide inhibitor attached to the active site of enzyme and undergoes a catalysis where it forms a product which is irreversibly or covalently binds with the enzyme and by forming a stable enzyme product complex. This alters the active site of the enzyme, and that is how this particular enzyme is got damaged irreversibly. So aromatase inhibitors. So how aromatase inhibitors works basically? So aromatase is an enzyme which acts on the androstenedione or testosterones to form the estrone and estradiols. So the aromatase inhibitors block this conversion of this particular estrone and estradiol. So estrone goes to the ER activation and leads to the proliferation and mutated cell and which leads to the cancer. And this estradiol generation of E2 and 3 to 4 quinone and deals with the leads to the DNA alkylation and depurination, and which also leads to the cancer. So this aromatase enzyme, so this is the active site of aromatase enzyme which has the iron 2 bind with the cysteine. Okay, and there is a 4-member nitrogen containing as a core, and the first step is the oxidation of this iron 2 which makes it iron 3. So iron 2 has a size which is higher greater than iron 3. Because of this large size, it cannot be fit in this core, so it goes up and forms this dome shape. Once it is in iron 3 position, the size got reduced and it now fits into the particular active site, and there is the oxygen which binds as a radical. And then we have this we have first one electron transfer to this radical and which forms a negative charge. This attacks on this particular androgens, and that's how this method of action it happens. Okay, you do not need to remember the all the molecules how the reaction happens, but remember that in the aromatase enzyme there is an iron core in the active site, and what is the basic active site structure, you need to remember this one. Okay, so the method of action of aromatase inhibitor is indeed is the suicide inhibition. Very good. Now I will go to the next problem. So problem number 9, it says that what is the general term used to indicate the growth of a new blood cells? Your options are number A biosynthesis, number B apoptosis, number C embryogenesis, and number D metastasis. What is the general term used to indicate the growth of the new blood vessels? Biosynthesis, apoptosis, angiogenesis, and metastasis. Let me know in the chat box A, B, C, or D which of the following you think is the correct answer. I want all of you who are present in the class, all of you to answer. Even if your answer is wrong, it is fine because it's only the practice session. But answering is very important. Otherwise, I won't understand that you're following the class or not. So what is the general term used to indicate the growth of the new blood vessels? Biosynthesis, apoptosis, angiogenesis, metastasis. What is the general term used to indicate the growth of the new blood vessels? Let me check the chat box. Okay, no answer till now. Okay, it's fine. Let me show you the correct answer and then let us discuss about it. So, the general term which is used to indicate the growth of the new blood vessels is number C, angiogenesis. So, angiogenesis, it is the term which used to indicate the new blood vessels. So, as the tumor grows, the cancerous cells require a steady supply of amino acids, nucleic acid bases, carbohydrates, oxygen, and growth factors if they are to continue multiplying. This means that the tumor has to have a very good blood supply. As the tumor grows in size, however, the cells become increasingly remote from the blood supply and become starved with these resources. Oxygen levels are also fall, resulting in a state of the hypoxia. This is particularly true for the cells in the center of the tumors. So, as a result of this hypoxia, inducible factors such as the HIF1, which is the hypoxia inducible factor 1, starts to build up within the tumor cells, and these factors serve to upregulate the genes that promote survival oxygen-related environments. For example, growth factors are released from the cell, such as the vascular endothelial growth factor or VEGF and the fibroblast growth factor or FGF2, which interact with the receptors in the endothelial cells nearby blood vessels and stimulate the cells to divide, leading to branching or extension capillaries, a process known as the angiogenesis. This is a particular angiogenesis process. Okay, this is the tumor cell. Once there is a short of the blood supply at the center of it, it activates these factors which are the VEGF and FG2, which which promotes these endothelial cells in synthesizing new blood vessels, and then angiostatin, thromboblastin, and matrix metalloprotease. These are activated, and this leads to the new blood metastasis of the blood cells, and which leads to the angiogenesis or the process of formation of new blood cells. The molecular mechanism of angiogenesis. The angiogenesis is defined as the formation of new capillaries from the pre-existing vasculature by migration of the proliferation of the endothelial cells. Okay, so this is the tumor cells, and this is the epithelial cells. As you can see, these are the epithelial cells, these are the tumor cells, and this bluish one, these are the supporting cells, and these are the platelets in the blood, and these are the soluble proteases basement membrane. These are the extracellular matrices. These are the here it also are written. Okay, so these are the intrains. These are the PGGF and activators. Okay, so angiogenesis was the correct answer. Now I will go to the next problem. So problem number 10 says etoposide is, your options are number A, intercalating topoisomerase poison, number B, non-intercalating topoisomerase poison, number C, intercalating topoisomerase inhibitor, and number D, non-intercalating topoisomerase inhibitor. This is the structure of etoposide. Okay, this is the structure of etoposide. So you have to let me know that etoposide is a which kind of which kind of agent. Your options are number A, intercalating topoisomerase poison, number B, intercalating topoisomerase poison, non-intercalating topoisomerase poison, number C, intercalating topoisomerase inhibitor, and number D, non-intercalating topoisomerase inhibitor. Let me know in the chat box A, B, C, or D which of the following is the correct answer. Let me check the chat box. Okay, I have one answer which says it is number B. Let me check if number B is the correct answer or not. So etoposide is, it is, yes, number B is the correct one. It is a non-intercalating topoisomerase poison. So now let us look at some non-intercalating topoisomerase poison. The anti-cancer agent etoposide and teniposide, they belong to a class of the compounds known as the podophylotoxins. So these are the semi-synthetic derivative of epipodophylotoxin. There's an isomer of the naturally occurring agent called the podophylotoxin. So now both of these agents act as a topoisomerase poisons. DNA strand breakage by this class of molecules is thought to occur by free radical process that involves 4' phenolic group. And what happens or what is proposed to happen is that this 4' phenolic groups, this one, okay. So this 4' phenolic group, this group forms a semiquinone radical, which is then generates again reactive oxygen species. Evidence for this comes from when you remove this or remove the, or if you remove this or derive this oxygen with the methyl, then the compound becomes inactive. The presence of the glucoside sugar moiety also increases the ability of molecule to induce the brakes. This glucoside sugar moiety increases the ability of the molecule to induce the brakes, and all. And this etoposide has this 4' phenolic group which forms a semi-hydroquinone radical, and this also initiates this. This is acts as an anti-cancer. I mean, acts as a topoisomerase poison. And if you have to identify this particular method of action, what you have to do is you have to derive this etoposide by methylating it in this position. Once it is methylated, this OMe group cannot form the semiquinone radical, and that is how this compound became inactive. Okay. So this is the non-intercalating topoisomerase poison. It is not intercalating like others which we have discussed earlier. It is a non-intercalating topoisomerase poison. Okay. Now I will go to the next problem. So problem number 11 says, which of the following reaction is involved in the method of action of calcimicin? Your options are number A, Balli-Silman reaction, number B, Suzuki coupling reaction, number C, Diels-Alder reaction, number D, Bergman cyclization reaction. Which of the following reaction is involved in the method of action of calcimicin? Balli-Silman reaction, Suzuki coupling reaction, Diels-Alder reaction, and Bergman cyclization reaction. A, B, C, or D? Which of the following is the correct answer for problem number 11? Which reaction is involved in the method of action of calcimicin? Let me know. Let me know in the chat box. A, B, C, or D? Which of the following is the correct answer? Which reaction is involved in the method of action of calcimicin? Balli-Silman reaction, Suzuki coupling reaction, Diels-Alder reaction, Bergman cyclization reaction. Okay, let me check the chat box now. Okay, I have an answer which says it is number D. Let me check if number D is the correct answer or not. So the correct answer for problem number 11, the reaction which is involved in the method of action of calcimicin is, very good, it's number D, Bergman cyclization. So first let us understand what is the mechanism of Bergman cyclization. So this Bergman cyclization happens mainly in the N-diene system. N-diene means N means that is double bond, and Y-ine means Y-N-E. This means a triple bond, and diene means a triple bond structure which happens twice. So here we have a triple bond followed by a single bond, double bond conjugation, and then we have another single bond, triple bond. So this is known as the end-die-in functionality. So when there is a heat or H nu means light source, so what happens? This forms this triple bonds. So one pi bond of this triple bonds, they form the radicals, and these radicals they actually forms this kind of 1,4 benzyl diol radical diradical. Okay, and this diradical can take up a proton from the second this kind of a molecule. This is the cycloaromatization reaction, and now this taking up a proton from a single molecule. Now this forms this benzene from this cycloaromatization, and we have this particular ring which has with this known as the Boltzmann cyclization. So in the mechanism, now let us understand the method of action of calciumycin. So this is the structure of the calciumycins. Okay, I have this. There is a spelling mistake here. Let me just write it up. Yes, so this is the structure of calciumycin. This is a particular we have this kind of endyne system here also, and here inside the site we have this sulfur moieties, and first step is the attack of the nucleophile in the second sulfur, and we have a thiol which is free now. Then the second step, this thiol attacks on this position, and we have this kind of attack this double bond position. This double bond goes up here, and we have a negative charge on oxygen. Now this particular negative this particular negative charge is stabilized by this group, and then we have this Boltzmann cyclization. Okay, now what happens during the Boltzmann cyclization? This particular endyne system forms this di 1,4 diradical and a phenyl ring, and this di 1,4 diradical attacks with the DNA, attacks DNA, and from DNA diradical. Okay, here you have seen that there is a phenyl cyclohexyl ion system. So I mean the two double bonds here, but here the DNA acts as a substrate, and this diradical of this number C complex, it attacks on the DNA and forms the DNA diradical, and this DNA diradical leads to the DNA double strand cleavage with the help of oxygen, and we have this DNA double bond strand cleavage. It acts as a lethal for the lethal DNA damage. Okay, so that is how this method of action of calciumycin needs the Boltzmann cyclization to happen to form this biradical which attacks on DNA and performs this DNA breakage. Okay, so correct answer was number D. Now I will go to the problem number 12, which says that which of the following heterocyclic rings is present in cimetidine? Your options are number A imidazole, number B pyrazole, number C indole, number D benzimidazole. Which of the following heterocyclic rings is present in cimetidine? Imidazole, pyrazole, indole, benzimidazole. Which of the following heterocyclic rings is present in cimetidine? Imidazole, pyrazole, indole, benzimidazole. Let me check the chat box now. Okay, I have one answer which says it's number C. Let me check if number C is the correct answer or not. So the heterocyclic ring that is present in cimetidine is it's actually number B, which is pyrazole ring. So first I have told it earlier, I think one time, that example of different nitrogen containing heterocyclic compounds and these are very important. Okay, especially nitrogen containing heterocycles are present in many biological systems. So remembering the names are very important. Also we have oxygen and sulfur, but it's the typically less number. Like we have first the 5-member heterocyclic rings. If it has oxygen, it's known as furan. If it has nitrogen, it's known as the pyrrole, and if it has the sulfur, it's known as the thiophene. Then we have the 6-member ring containing one nitrogen. It is known as pyridine. If this is known as indole, then this is known as benzofuran. This is known as the carbazole. This is known as quinone. This is known as quinoline. This is known as isoquinoline. This is known as the imidazole. This is known as oxazole. This is as pyrazole. So in imidazole, two nitrogen are in 1,3 position. In pyrazole, two nitrogen are 1,2 position. In oxazole, one nitrogen and one oxygen are present in 1,3 position. We have pyridazine, which is a 6-member ring having two nitrogen in 1,2 position. We have pyrimidine, which has two nitrogen, pyrimidine, which has two nitrogen in 1,2 position, and we have purine, which has two, one 6-member and one 5-member ring joined with each other, and the nitrogens are present in 1 and 3 position of each ring. So the cimetidine, this is the structure of cimetidine. Okay, this is the cimetidine. Okay. So the synthesis of the cimetidine. Okay. Sorry, I think I have made a mistake. 1 second. Okay, so I have made a mistake here. Sorry for that. The cimetidine. This does not have this pyrazole, but it has a imidazole ring. Okay, I have made a mistake. Imidazole. Okay. So the correct answer was number A, imidazole ring, not pyrazole ring. It was my mistake. Sorry. Because as you can see, the structure of the cimetidine, it has a 5-member ring where we have the nitrogen in 1 and 3 position. Okay, so this cimetidine has a imidazole ring, not a pyrazole ring. Okay, because in pyrazole ring the nitrogens are 1, 2 position. So this has a imidazole. So correct answer is number A. So was your answer number A? I am not sure that there was one answer. So was it number? No, it was number C. Okay, fine. So the correct answer is number A, which is the imidazole. Imidazole is present in cimetidine. Now let us see what is the synthesis of cimetidine. So the synthesis of cimetidine was originally carried out as a 4-step process where the lithium aluminium hydride was used as a reagent for the initial reduction step. Subsequent research revealed that the reduction could be carried out more cheaply and safely using sodium in liquid ammonia. So it become method of use in the manufacturing process. Okay. So the first step is the reduction of this ester group to the alcohol group. Okay. And C double bond COET, it converts CH2OH. This reduction can be done by lithium aluminum hydride or sodium in liquid amine and tertiary butanol, and also in the methanol and NH4Cl. Okay. Then followed by we have this thiol, which attacks on this alcohol and forms this compound, then followed by the reaction with this cyanide, which makes this portion at opposite portion, then at the last with the methyl CH3NH2, which makes this SME to NHME. Okay, so this is the cimetidine structure. So biological activity, cimetidine inhibits the gastric acid release by acting as an antagonist of H2 receptor. It does not show any toxic side effects observed for metimide and has been shown to be slightly more active. It has been found to inhibit the pentagastrin from stimulating release of the gastric acid. Pentagastrin is an analog of agastrin, and the fact that cimetidine inhibits it suggests that some relationship between histamine and gastrin in the release of gastric acid. Cimetidine was first marketed in UK in 1976 under the trade name of the Tegamet and derived from antagonist and cimetidine. It was first really effective anti-ulcer drug, doing away the need to surgery for several years. It was best selling prescription medicine until it was pushed to second place in 1988 by ranitidine. Okay. Now I will go to the next problem. So problem number 13 says, which one of the following activities occurs following the stimulation of H2 receptors? Your options are number A, vasodilation, number B, uterine contraction, number C, bronchial smooth muscle contraction, number C, enhanced secretion of hydrochloric acid in stomach. Which of the following activities occurs following the stimulation of H2 receptors? Vasodilation, uterine contraction, bronchial smooth muscle contraction, enhanced secretion of hydrochloric acid in the stomach. Which of the following activities occurs following the stimulation of H2 receptors? Vasodilation, uterine contraction, bronchial smooth muscle contraction, enhanced secretion of hydrochloric acid in stomach. A, B, C or D, let me know the correct answer in the chat box. Which one of the following activities occurs following the stimulation of H2 receptors? Let me check the chat box. Okay, I have one answer which says it's number A. Let me check if number A is the correct answer or it's not. So, the correct answer is it's number D, enhanced secretion of hydrochloric acid in stomach. It is the activity which occurs following the stimulation of H2 receptors. So H2 receptors, or it's also a part of a histamine receptor. So H1, H2, H3, and most recently H4 are the different histamine receptors. These are the G protein coupled receptors. Activation of H1 causes the contraction of smooth muscles in gut, uterus, and bronchi. Contraction of the bronchi leads to asthma. Stimulation of H1 receptors or smooth muscles in the blood vessels cause muscle relaxation for resulting vasodilation, may result in the severe blood fall in the blood pressure. In CNS, H1 and H2 receptors predominantly localized on the presynaptic membrane. H3 receptors appear in the function in the presynaptic receptors of the histamine autocorrector and some peripheral organs. H4 receptor restricted to the intestinal tissue, spleen, immune system, T cells, therapeutic of H4 blocker in allergic and immunitary diseases. Now H2 and H1 receptors are most in common. So the definition is H1 are the histamine receptors that couple with the GQ11 stimulating phosphodiesterase C, and H2 receptors are histamine receptor that interacts with the GS to activate adenylyl cyclase. Location periphery of the body, adrenal medulla, vascular endothelium, heart, central nervous system. H2 receptors are present in the central nervous system and the peripheral periantal cells located in the stomach. The antagonists are the diphenylhydramine, loratadine, cetirizine. These are the antagonists of H1 receptors. Ranitidine, cimetidine, famotidine, and nizatidine are the H2 receptors antagonists. The function of H1 receptor is the ileum contraction, modulate the circadian cycle, itching, systemic vasodilation, and bronchocontraction or allergy-induced asthma, and speed up the sinus rhythm, stimulation of gastric acid secretion, smooth muscle relaxation, inhibit antibody synthesis, T cell proliferation, cytokine production. Okay, so enhanced secretion of the hydrochloric acid in stomach is occurs due to the H2 receptor stimulation. Now I will go to the next question. So problem number 14 says anti-ulcer drug omeprazole is, your options are number A, reversible proton pump inhibitor, number B, antagonist of H plus K plus ATPase, number C, agonist of H plus K plus ATPase, number D, irreversible proton pump inhibitor. So anti-cancer, anti-ulcer drug omeprazole is which kind of agent? Number A, reversible proton pump inhibitor. Number B, antagonist of H plus K plus ATPase. Number C, antagonist of H plus K plus ATPase. Number D, irreversible proton pump inhibitor. In the chat box, let me know A, B, C or D which of the following is the correct answer. Okay, let me check the chat box now. I think I have one answer which says it's number B. Let me check if number B is the correct answer or not. So anti-ulcer drug omeprazole is, it is a irreversible proton pump inhibitor. The correct answer is number D, irreversible proton pump inhibitor. This is the structure of omeprazole. This is the benzimidazole ring is present here, and also a pyridine ring is also present in the omeprazole. So the proton pump is the only present in the canalicular membrane of the parietal cells, and the crucial for the mechanism by which the hydrochloric acid is released into the stomach. It is called the H plus K plus ATPase because it pumps protons out of the cell into the canalias. At the same time, it pumps the potassium ions back in. This energy is required for this process as both protons and potassium ions are being moved against the concentration gradients. In fact, the ratio of protons inside the cell to protons in the canaliculus is 1 to 10 to the power 6. The energy required to carry out this exchange is obtained by the hydrolysis of ATP. Hence, this term is the ATPase. The pump is not responsible for the efflux of the chloride ions. This depart from the cell through the separate chloride ion channels. The outflow closely matches with the efflux of the protons, such as the chloride ion is released in every proton is being pumped out. As a result, hydrochloric acid is formed in the canaliculus rather than inside the parietal cell. As each chloride ion departs from the cell, they are accompanied by the potassium ion, which flows through the own ion channel. No energy is required for this outflow because the potassium ion is flowing down in a concentration gradient. Potassium ion acts as a counter ion in the chloride ion, and once the canaliculus is pumped back to the cell by proton pump described previously. Consequently, potassium ions undergo a cyclic movement in and out to the cell. So is the role of the proton pump in secreting the hydrochloric acid. This chart shows it here. And there are 4 PPIs or proton pump inhibitors in the clinical use: omeprazole, lansoprazole, pantoprazole, and rabeprazole. The S enantiomer of omeprazole has been also approved recently. All the PPIs have this pyridinyl methyl sulfonyl benzimidazole skeleton. This is pyridinyl methyl sulfonyl benzimidazole skeleton and acts as a product since they are activated. Now, when they reach the acidic canaliculi of the parietal cells, once activated, they bind irreversibly to the exposed cystine residues of proton pump and block the pump, preventing further release of hydrochloric acid. There is a big strategic advantage in the inhibiting the proton pump rather than blocking the histamine or cholinergic receptors. For example, H2 antagonists block the histamine receptors and block the stimulatory effect of the histamine, but it does not block the receptors of acetylcholine and gastrin, and so it is still possible for the parietal cells to be activated toward the secretion. The proton pump is downstream of these targets and operates the final stage of hydrochloric acid release. Blocking it prevents the release of the hydrochloric acid regardless of what the mechanisms are involved in the stimulating hydrochloric acid secretion. Okay. Now I will go to the next problem. So problem number 15 says, what is the correct order of intermediates in the route of the cholesterol biosynthesis? Your options are number A, HMG-CoA, then followed by mevalonic acid, followed by squalene. Then squalene, the number B is squalene, followed by HMG-CoA, followed by 7-dehydrocholesterol. Number C, mevalonic acid, followed by farnesyl diphosphate, followed by squalene. And number D, acetyl-CoA, followed by HMG-CoA, followed by squalene. What is the correct order of intermediates in the route of the cholesterol biosynthesis? What is the correct order of intermediates in the route of cholesterol biosynthesis? Your options are number A, HMG-CoA, mevalonic acid, followed by squalene. Then squalene, HMG-CoA, dehydrocholesterol. Number C, mevalonic acid, farnesyl diphosphate, squalene. Number C, acetyl-CoA, HMG-CoA, or squalene. What is the correct order? You have to identify the order. Let me check the chat box. Okay. Okay, let me show you the correct answer for problem number 15. So the correct order of the intermediates in the route of the cholesterol biosynthesis is, it is number C, mevalonic acid followed by farnesyl diphosphate followed by squalene. So first step, starting point of the cholesterol biosynthesis is acetyl-CoA. Okay, remember it very well. The first is the acetyl-CoA followed by acetoacetyl-CoA, and the enzyme is the acetoacetyl-CoA thiolase. Then another acetoacetyl-CoA comes, and we have the HMG-CoA reductase CoA synthesis, which forms the 3-hydroxy 3-methyl-glutaryl-CoA or HMG-CoA. Then HMG-CoA reductase forms mevalonic acid, which forms farnesyl diphosphate and forms squalene. By this farnesyl diphosphate to squalene conversion happens through the squalene synthesis. Then the squalene forms a 7-dehydrocholesterol intermediate, which goes up to the cholesterol by the action of the enzyme 7-dehydrocholesterol delta 7 reductase. Okay. So the correct order is mevalonic acid followed by farnesyl diphosphate followed by squalene. Now I will go to the next problem. Problem number 16 says statins work as, you have to identify, number A, HMG-CoA synthetase inhibitor, number B, HMG-CoA reductase inhibitor, number C, squalene synthetase inhibitor, and number D, acetoacetyl-CoA thiolase inhibitor. Statins work as which kind of inhibitor? HMG-CoA synthetase inhibitor, HMG-CoA reductase inhibitor, squalene synthetase inhibitor, or acetoacetyl-CoA thiolase inhibitor. Statin works as, number A, HMG-CoA synthetase inhibitor, number B, HMG-CoA reductase inhibitor, number C, squalene synthetase inhibitor, number D, acetoacetyl-CoA thiolase inhibitor. Let me check the chat box. Okay. Okay, fine. Let me show you what is the correct answer for this problem. So, statins works as, number B, HMG-CoA reductase inhibitor. Okay. So HMG-CoA reductase inhibitors, also known as statins, are a class of lipid-lowering drugs that reduce the illness and mortality in those who are at high risk of cardiovascular disease. They are the most common cholesterol-lowering drugs. Reduce the risks of the illness related to the atherosclerosis with a varying degree of the effect depending on the underlying risk factors and history of cardiovascular disease. Clinical practice guidelines generally recommend the people to start with the lifestyle modification through a cholesterol-lowering diet and physical exercise. The mechanism, the HMG-CoA reductase pathway, which is blocked by the statins via inhibiting the rate-limiting enzyme HMG-CoA reductase. Statins act as competitively inhibiting the HMG-CoA reductase and the rate-limiting enzyme of the mevalonate pathway. Because statins are similar in the structure of HMG-CoA on molecular level, they will fit into the enzyme's active site and compete with the native substrate HMG-CoA. This competition reduces the rate in which the HMG-CoA reductase is also is able to produce the mevalonate. The next molecule is the cascade that eventually produces cholesterol. So these are the different statins, which are very good class of the lipid-lowering drugs. We have the pravastatin, simvastatin, fluvastatin. Atorvastatin is a very important compound or very important lipid-lowering drug. Rovastatin is also very important drug. Pitavastatin, mevastatin, avostatin. These. So the green ones are the FDA approved. Okay, the year of the FDA approval. The green the years. and the red one is the pharmacophoric group. These portions are the pharmacophoric group of the each compounds. Okay. Now I will go to the next problem. Problem 17 says, which of the following is not a third generation aromatase inhibitor? Your options are number A, anastrozole, number B, letrozole, number C, vorozole, and number D, fadrozole. Which of the following is not a third generation aromatase inhibitor? Anastrozole, letrozole, vorozole, and fadrozole. Which of the following is not a third generation aromatase inhibitor? Anastrozole, letrozole, vorozole, fadrozole. Let me check the chat box. Okay, fine. Let me show you the correct answer. So, the compound which is not a third generation aromatase inhibitor is number D, fadrozole. So generation of different aromatase inhibitors. So there are two type of aromatase inhibitor. One is a steroidal inhibitor and another is a non-steroidal inhibitor. The first generation steroidal inhibitor is none, but the type 2 or non-steroidal inhibitor of first generation is amino glutathione. Then the second generation or type 1 steroidal inhibitor is formestin, and the type 2 non-steroidal inhibitor of the second generation is fadrozole and roglutamide, and the third generation is the exemestane, which is the aromacin for the type 1 steroidal inhibitor, and anastrozole, letrozole, vorozole. These are the third generation non-steroidal or type 2 inhibitor. So aromatase inhibitors and inactivators, mechanism of action, steroidal, the examples I have told you earlier. The steroidal inhibitors, they bind with the substrate binding site of the aromatase, and the non-steroidals bind to the heme part of the aromatase. I have already shown you, so there is a heme part in the aromatase, there is an iron center, and also this the irreversible binding to enzyme is a steroidal one, and the is non-steroidal was the irreversible binding of the enzyme. Okay. And this is the structure of the letrozole and anastrozole, which are the very famous and very most important aromatase inhibitors, which are FDA approved drugs also. So these triazole 1,2,4 triazole portion, it interacts with the heme portion of the aromatase enzyme. Okay. Now, next problem. Problem number 18 says, which of the following is not a QSR parameter? Your options are number A, hydrophobicity, number B, hydrophilicity, number C, steric, and number D, electronic. Which of the following is not a QSR parameter? Which of the following is not a QSR parameter? Hydrophobicity, hydrophilicity, steric, or electronic? Let me check the chat box now. I have one answer which says it's number A. Okay, let me check if number A is the correct answer or not. So, the factor which is not a QSR parameter is, it is number B. Hydrophilicity is not a QSR parameter. So what is QSR? A quantitative structure activity relationship, or QSR is a mathematical relationship which correlates the measurable or calculate the molecular properties to some specific biological activity in terms of an equation. QSR attempts to find consistent relationship between the biological activity and the molecular properties so that these rules can be used to evaluate the activity of new compounds. Understanding QSR regarding electronic effects, steric effects, and lipophilicity. Physicochemical parameters. Various parameters used in QSR are hydrophobicity, partition coefficient, pi substituent constant, steric parameter, Tufts constant, Verloc steric parameter, electronic parameter, Hammett constant, and dipole moment. Okay, so these are the different parameters of the QSAR, and how to it partition coefficient of substitution constant. Okay. Now next problem. Problem number 19 says camptothecane or CPT works as number A, topoisomerase 1 inhibitor, number B, topoisomerase 2 inhibitor, number C, DNA guidase inhibitor, and number D, DNA polymerase inhibitor. Camptothecane or CPT works as which type of inhibitor? Topoisomerase 1, topoisomerase 2, DNA guidase, or DNA polymerase? Camptothecane or CPT works as which kind of inhibitor? Topoisomerase 1 inhibitor, topoisomerase 2 inhibitor, DNA guidase inhibitor, DNA polymerase inhibitor. Camptothecane or CPT works as which kind of inhibitor? Topoisomerase 1 inhibitor, topoisomerase 2 inhibitor, DNA guidase inhibitor, DNA polymerase inhibitor. Let me check the chat box now. Okay, I have one answer. Oh, okay, I have two answers, one number B and one number A. Let me check which of the following is the correct answer. So camptothecin or CPT works as a number A, topoisomerase 1 inhibitor. So camptothecin, CPT, is a plant alkaloid. It is the most established inhibitor of DNA topoisomerase 1. Camptothecin is pharmacologically unique because unique topoisomerase 1 is the only target of this drug, and CPT binds neither to TOP1 alone nor the DNA alone, but only to TOP1 DNA cleavable complex. So the next class of important molecule that we have to look is the camptothecin. Camptothecin is a natural product that was extracted from the Chinese bush Camptothecea acuminata in the 1966. Camptothecin stabilizes the cleavable complex that is formed between the DNA and the enzyme topoisomerase 1. So by doing this, what happens that it results in the single strand breaks to accumulate DNA. You may also remember the topoisomerase 1 and 2 both need the DNA for their function. The difference between topoisomerase 1 and topoisomerase 2 is that topoisomerase 2 cleaves only one strand of DNA, whereas topoisomerase 1 cleaves one. Okay, so it will be 1. Okay, there is a printing mistake. And topoisomerase 2 cuts the both strands of the DNA. Since the complex between DNA enzyme topoisomerase 1 is stabilized single strand break and does not interfere and accumulates the DNA, resulting in the inhibition and proliferation. These single strand breaks can be repaired if the drug departs, but if the replication taking place when the drug enzyme the DNA complex is present, an irreversible double strand break takes place, which leads to the cell death. Semisynthetic analogs of camptothecin have been developed as a clinical useful anti-cancer agents. Okay. So yes, camptothecin is a type 1 inhibitor of the topoisomerase or topoisomerase 1 inhibitor. Now last question of today's session, which says for the better bioavailability of the drug, what should be the maximum number of rotational bonds? Your options are number A 10, number B 7, number C 5 and number D 13. For better bioavailability of a drug, what should be the maximum number of the rotational bonds? 10, 7, 5, 13. For better bioavailability, what should be the maximum number of rotational bonds? 10, 7, 5, 13. What is for better viability of the drug? What should be the maximum number of rotational bonds? Let me check the chat box. Yeah, I have two answers. Both are saying that it is number B. Let me check if number B is the correct answer or not. The correct answer is yes, it's number B, 7. 7 should be the maximum number of rotational bond for better viability of a drug. So, a rotable bond is defined as any single non-ring bond. Okay, a single bond which is not of the non-ring, which is not present in the ring, attached with a non-terminal non-hydrogen atom. Okay, it should not be non-terminal, it should be non-hydrogen. And so amide CN bonds are not accounted because they are high barrier of rotation. So amide CN bond has a, this bond of amide CN is has a high rotation barrier, so it does not count as a single strand bond. So if, as you can see, this compound is atenolol. Okay, it has 1, 2, 3, 4, 5, 6, 7, 8 rotational bond, and the viability is 50%. Whereas this propanolol, which is a which is another just a variation of atenolol, this which has a 1, 2, 3, 4, 5, 6, 6 rotational bond. So as it has a number 6 less than number 7, its viability is greater than 90%, whereas atenolol, which has the more than 7 rotational bond, it has a viability of only 50%. Okay, so the maximum number of rotational bond for the having a better viability of a drug, it should not cross the number 7. So this was the last question of today's session. So today what we discussed, we mainly discussed about various anti-cancer drugs, various targets of the anti-cancer drug. We discussed about aromatase enzyme, the aromatase inhibitors, different compounds. Then we discussed about the proton pump inhibitors and anti-ulcer drugs. We discussed a little bit about the cholesterol biosynthesis and the cholesterol lowering drugs also. So these topics were your last topics for your course Organic Chemistry and Biology and Drug Development. And today in our week 11 session, we concluded the whole complete course and the last, last topics of this course of Organic Chemistry and Biology and Drug Development. Now I will share my email ID with you here. So this is my email ID. If you have any doubt, any query, you can let me know through an email. So we will have two more classes. Okay, one is our last live session class of Monday. In our Monday, our regular classes held from happen from 6 p.m. to 8 p.m. So which will be on next Monday. That will be our last Monday class, and it will be a summary class. It will not only it will be a summary class. I will just try to see. I will check if you have remember all the course. So please try to revise the course, because I will give you around 10 to 12 questions related to the complete course, which you have to solve start from the week 1 to week 12, and followed by I will discuss few topics which I couldn't discuss earlier because of the time limit. These topics are not so much involved in your course, but it will be beneficial for you for this particular topics of organic chemistry and biology and drug development. And the last class we will have on 16th of October, which is Wednesday. It will be notified to you later also, and we will have a 1 hour class from I think 7 p.m. to 8 p.m. As such the timings are. So that 1 hour summary class I will only discuss the topics which are important for your examination purpose. Okay, so do not miss the last two summary lectures, which one is on 14th October from 6 p.m. to 8 p.m. and another on 16th October from 7 to 8 p.m. You will be reminded through the email also. So let me know if you have any doubts, any query, or you want me to tell you some topics in your last classes, the particular topics which you find. So it will be the exam date. It will be notified to you via the email. Okay, in the SWAM portal it will be updated, and via email it will be communicated to you soon. The exam date and all the things related to the examination. Okay. So I'm closing the session for today. If you have any query, any doubt, please email me or in this email ID. Okay. Thank you all for joining. I'm closing the session for today.